to adapt this quality first transmission from human to human. One, most probably through respiratory route, it will be very easy. Third, it can mutate and change the structure so that it can escape the immune system of human, and it can cause severe disease. It may cause mild disease. ऐसा नहीं कि सारे पैंडेमिक सीवियर ही होते हैं. इन्फ्लुएंजा तो हमारे यहाँ होता ही है ना, सीजनल इन्फ्लुएंजा आप जानते हो. सभी को सर्दी खांसी जुकाम बुखार होता ही है, आपको भी होता होगा, लेकिन कभी we never bothered about this influenza. But suddenly the COVID came and we are very scary about the COVID and we just hide ourselves in the home. And everybody was so scared because its fatality rate was very high. So similarly, such incidences occur in the past also. 100 years ago, there were almost 50 million people were killed by the influenza virus pandemic. So it changes. So minor changes. अभी आप देख रहे हो कोरोना जैसे हो रहा है, तो कोरोना में there are so many variants, है ना? अल्फा है, बीटा है, डेल्टा है, अभी आपने देखा होगा डेल्टा प्लस बी. So these are the minor changes occur in the structure or antigenic nature of the organism, which is not totally unrelated to the previous one, but it is different. But in case this is known as antigenic drift, these are the minor changes which can leads to some variant of the, but the originality of the organism does not change. But if there is some genetic change in the virus and it totally changes its nature of the antigen, then it will be totally a new antigen. Right? So it's a drastic change and it is basically related to the gene. There is some extra gene or some gene is modified. There is a replacement of some gene or some introduction of some gene in the virus, which makes the virus totally different from the previous one. This is known as antigenic shift. The antigen which formed by this change will be entirely different from the previous one, and the organism is totally unrelated to the previous one. फिर ये किसी को नहीं मानेगा, कोई पिछली कोई वैक्सीन नहीं, कोई एंटीबॉडी नहीं, कुछ नहीं. This is known as antigenic shift. So gradual change is known as drift and sudden change is known as shift. So shift usually occur, it takes around 10 to 15 years to shift the genetic structure. So influenza may we have noticed that every 10 year it changes. Genetic shift occur every 10 year, right? अब ये कोरोना चल रहा है, तो कोरोना में भी ऐसे ही शिफ्ट हर 10 साल के बाद आए, पता नहीं 5 साल के बाद आए, तो 5 साल के बाद में हमें नया थ्रेट होगा. डिपेंडिंग अपॉन द, अभी तो हमें पता ही नहीं कुछ भी, ये तो हमने पिछले 100 साल का इन्फ्लुएंजा को देखा है, तो इसलिए we are able to tell you the trends of this virus. But so we have to see the trends of corona also, the similar. Right. So antigenic shift and antigenic drift, these are the mainly responsible for variation and the total change in the structure. So drift, remember the word drift and shift, right? Now we'll see how they are classified. And second, one of the important another thing is that the influenza virus, there are various types. They are divided into 4 types: A, B, C, and D. Right? Type A can cause infection in bird, that is avian flu. It can also cause infection in pigs, that is swine flu. It can also cause infection in human, that is human flu, right? So type A is a very dangerous virus because it can cause infection in birds, animals, and human, right? Now these viruses, अभी आपने पहले general virology पढ़ा होगा, तो what whenever the virus enter, तो it has to first step is the adsorption, then penetration, है ना, uncoating. These are the steps. The first step is the adsorption. तो receptor होगा तभी तो वो adsorb होगा. So avian birds and there are receptors present on the human cell. There are receptors on the avian and there are receptors on the swine, means pig, respiratory epithelium. But usually what happens, the receptor for avian flu is very less and very few in number in human. So avian flu directly transmitted to human is usually not possible. तो जनरली जो बर्ड फ्लू आपने नाम सुना होगा बर्ड फ्लू तो बर्ड फ्लू इज अ epidemic disease in the bird which kills the bird but ये receptor नहीं रहने के कारण this virus does not affect the human. ऐसा नहीं है कि वो अडेप्ट नहीं कर सकता, हो सकता है थोड़े दिन बाद अडेप्ट कर ले and then it can attack the human also, but the receptors are very few. So these infections, human infections, are seen with the avian flu, but only in cases who are in close contact with the birds. For example, poultry workers हैं जो कि रोज poultry के contact में आते हैं. Only infections are seen in those workers, and human to human transmission was not seen. Right? So there were the outbreak of bird flu, and they are transmitted to human, but there are very few cases, and they are also with the close contact with the birds. Those workers which are involved in the poultry or in the animal farming, only those were involved, but not the normal human being, and human to human transmission was not there. Now there is a swine. Pig is a reservoir for. Pig is a very dangerous for the influenza. Pig is the reservoir. It has receptors for both avian as well as human flu. So pig can be affected by avian flu, human flu, and swine flu also. So if such type of infection, if suppose he get the infection from all the three simultaneously, sometimes what happens, there is a genetic mixing up of the organism occur. That is known as genetic reassortment. H1N1 का नाम सुना है आपने? स्वाइन फ्लू कहते हैं उसे, है ना? हाँ या ना में भी जवाब दे सकते हो. हेलो. चल रहा है. So swine flu का नाम सुना है? तो swine flu is one of the very dangerous virus that was evolved from the pig, and it's not a swine. We named it swine flu, but it's not. It is originated from the swine. But if you see, if we study the genetic structure of this virus H1N1, it has one part of avian flu virus, one part of human virus, and two part of swine flu virus. So it's a genetic reassortment occur inside the pig, and then from the pig it is transmitted to the human, and from the human it is person-to-person transmission. Right? So H1N1. So genetic reassortment or the mixing up of the genetic material from different animal strain is responsible for the pandemic disease. Okay, so this is in short about. Now we will see. So we have seen that in 1918 there was an influenza pandemic and it killed almost 50 million people, and especially in the young adult 20 to 40 years of age group, right? We also seen that it has a potential of highly contagious, rapidly evolving, and can cause epidemic, right? Now coming to myxovirus. Myxovirus, there are myxo means mucin. It has a these viruses. They have a receptor. They are attached to the receptor on the RBC, that is mucin, right? So that's why they are known as myxovirus. They have a receptor, especially on the RBCs and other cells also. So they are basically enveloped viruses, and they having ability to adsorb on the mucoprotein receptor on the erythrocyte, causing hemagglutination. This is the feature of. ऐसा नहीं है कि दूसरे सेल्स में नहीं होता, अदरवाइज तो रेस्पिरेटरी इपिथेलियम में तो होगा ही नहीं, ओनली आरबीसी है तो. बट hemagglutination is because once upon a time hemagglutination was the only test to diagnose the influenza infection. So hemagglutination, that's why they have given stress on the hemagglutination, and it has two peplomers, so hemagglutinin and neuraminidase, that we will see. So myxovirus, basically they are classified into orthomyxo and paramyxo. So orthomyxo is the influenza virus, whereas paramyxovirus include parainfluenza, mumps, measles, respiratory syncytial virus, Newcastle disease virus, and metapneumovirus, right? So these are the virus. So today we will see the influenza virus. Now, what is the difference between orthomyxo and paramyxo? So orthomyxo is a smaller virus. Size is around 80 to 20 nanometer, whereas paramyxo, they are bigger virus, slightly bigger, 100 to 300. Shape is spherical, and paramyxo virus, they are pleomorphic. Both are enveloped virus. The important one, they are marked in the red, genomic structure. Now see the genomic structure of orthomyxovirus, the segmented 8 piece of RNA. This is very important. It has a segmented 8 piece of RNA. Now each segment represents one antigenic structure. So any alteration in the gene, any segment, 2, 3 segments are replaced, then definitely the new antigen will be formed. Whereas the paramyxovirus, they have just a single linear RNA, so no ability to cause pandemic, no variation are seen. Genetic reassortment are common in orthomyxovirus, whereas paramyxovirus, it was not reported. Antigenic stability, so it is unstable, variable, keep on changing, whereas paramyxo, they remain stable. Site of synthesis of ribonucleoprotein is in the nucleus for orthomyxovirus, whereas it is in the cytoplasm. So these are the basic differences. And these differences, these 3 differences, that is the segmented 8 piece of RNA, gene assortment is common, and antigenic stability is highly antigenic variable, is the 3 features which makes him a potential or dangerous pathogen for that may lead to pandemic. So I told you about the A, B, C, D. The type A is very dangerous because it can cause infection in human, animal, birds, more severe in the elderly, and all the pandemics are because of type A. Type B occur only in the human, that is more common in children, normal flu. Type C, human only, mild infection, and type D, only animals. So These three are not important, but type A is very important. Now, uh, the type of the influenza, they are basically, first of all, I show you the structure. A and B may another difference. A and B are morphologically similar, but C has only 7 segments instead of 8, and lack of neuraminidase gene. One uh, genetic segment is absent in type C. That is not very important for you. Yes. Now this is the structure of influenza virus. On the basis of nucleoprotein and matrix protein, which is written in yellow. On the basis of nucleoprotein and the matrix protein, the influenza virus is classified into 4 types, that is A, B, C, D. But they are further A type is further subtyped on the basis of presence of peplomers or the spike protein on the cell surface of virus. Spike protein, they are of 2 types. One is known as hemagglutinin and other is known as neuraminidase. So it will be written here HA and NA. So there are 2 peplomers. These are known as peplomers. They are the projecting or the spike proteins. So on the basis of this spike protein, hemagglutinin and neuraminidase. Now, most changes occur here only. These peplomers are changed. So H1N1, H5N1, H3N2. So there are various variations you can see. H5N1. So we will see that. So variations occur and strain changes. Whether it will cause mild, moderate, depends. Right? Now this hemagglutinin is usually a triangular in shape. And as we have discussed that it has a receptor for the glycoprotein receptor on the RBC and attached to the RBC and cause hemagglutination. And other neuraminidase is the mushroom shaped structure, peplomer present on the cell surface. Now, what is the of this neuraminidase? This virus, once enter, it get absorbed through hemagglutinin on the cell surface, RBCs, right? This after getting infection, after infecting the RBC, after some time, this neuraminidase enzyme, it destroy the glycoprotein receptor to which the virus has attached, and it set free the virus to infect other RBCs, right? This process of setting free the virus to infect other RBCs or other cells is known as elution. So main function of this neuraminidase is to set free the virus attached, and it will facilitate the infection, to spread of infection to the other cells, right? Now the virus from which the cells from which this virus has just eluted, it will not get reinfected by the same strain, but it can be infected with the other strain. Same strain इसको affect नहीं कर पाएगा. So this process that you have to see. So अगर antibody हमने hemagglutinin के against में बनाई, so it will prevent the infection. It will help in the prevention of infection because it is required for the adsorption or the attachment of the virus on the cell surface. So अगर हमने antibody hemagglutinin के against में बनाएंगे, so there will be no attachment, so no infection. That will be preventive. If we make the antibody against neuraminidase. So how it will protect us? So even if you are getting infected, you are getting infected, then the spread of infection, the severity of infection will be less because it will prevent the illusion of the virus. So infection, spreading of infection will not occur, right? So both the antibodies are very important. That is same. Okay. Now influenza virus, there are 18 heme agglutinin subtypes and 6 are found H1, H2, H3, H5, H7, H9. Out of these 6, these are found in human. Out of these, H1 and H2 has a tendency to undergo a radical changes and cause pandemic. That is, they can antigenic shift can occur and can cause pandemic. Hemagglutination and hemagglutination inhibition. Hemagglutination and hemagglutination inhibition are the important diagnostic tool in the previously in past. Actually, now these tests are not done because better tests are available. That's why they have given much more importance to this uh, hemagglutination test. Now, neuraminidase. There are 11 subtypes of neuraminidase identified, but only N1 and N2 are from the isolated from the human. Right. The rest of the thing we have discussed that the same. They degrade the mucus layer, exposing the epithelium membrane. Infection facilitate the detachment, release of dissemination of newly found virus particles. Right. This is not important. Influenza virus multiplication, type specific. I told you that they are typed on the basis of matrix protein and ribonucleoprotein. Strain specific, they are on the basis of hemagglutinin and neuraminidase. We have discussed about it is a unique genetic shift and antigenic variation is the unique feature, and we have discussed that antigen, what is antigenic shift and what is antigenic drift. So I just show you the table which show the difference. See this diagram we have discussed also. This is the human flu. This is the human flu, swine flu. Both the virus infect the pig, and here the genetic reassortment occur, and this genetic reassortment leads to the formation of new virus. This is antigenic shift. Sometimes this bird flu, because pig is susceptible to avian strain also. So human and bird, avian, human, and swine flu simultaneously attack. They can infect pig, and in the pig genetic reassortment can occur. So pig is the reservoir host. So this is the genetic shift, how this mechanism operates. Right? There is a minor change in the uh, amino acid sequence, so that is a mutation or something. So this is the table which showed that what is antigenic shift and what is antigenic drift. Now, antigenic shift, I told you, it is abrupt, drastic, discontinuous variation in the antigenic structure. It's a totally different structure, and it's abrupt, drastic change. Whereas antigenic shift is a gradual, sequential, regular antigenic change at periodic interval. So it is not a drastic change, slow process, and the new strain is related to the previous one. The result is in the antigenic shift, this result in the new strain which is unrelated to the predecessors. Whereas in the antigenic drift, result in a new strain related to the, but it is related to the predecessor. Antigenic shift is mainly because of genetic or gene reassortment, whereas antigenic drift is due to mutation and selective pressure. अगर antibody आपने बढ़ा लिए, अगर सर्वाइवल प्रॉब्लम हो रही है वायरस को, उसने देखा थोड़ा सा मॉडिफिकेशन कर लिया ताकि he can escape the antibody. So selective pressure of antibody and mutation responsible for epidemic and pandemic. It can respond, drift can responsible for epidemic, but for pandemic, antigenic shift is required. Right? So this is the difference between. Now WHO has nomenclature. WHO and other agencies all in the world, they keep an eye on this progress of influenza. कौन सा influenza कहाँ पे चल रहा है, ये सब का एक WHO and other agencies, they keep an watch on this. कि कहीं epidemic या कहीं pandemic तो नहीं हो सकता है. Every time, whenever there is outbreak, they used to collect the sample and see. Yeah, so they are basically they are given names. तो name कैसे दिया जाता है, ये nomenclature भी एक बड़ा interesting है. According to WHO, the complete designation of the strain, which include type. Type means whether it is A, B, C or D. Usually it is A. Then host of origin. इसका origin कहाँ से था? Whether it was a bird, swine or human. Right? तो ह्यूमन का रहेगा तो हम नहीं इसको काउंट करेंगे, because we are study only in human. But if the origin is swine or avian, then we will mention it. Place of origin, कहाँ से ये start हुआ था? On first time, whenever this new strain they isolate it and give the serial number. तो कहाँ से ये first time isolate हुआ था, and when it was isolated, the serial number was given. तो serial number ताकि हमें पता चल सके कि ये सीरियल नंबर 1942 में भी हुआ था और ये अभी 2020 में भी हो रहा है. तो ये उस पर ट्रैक रखने के लिए. ईयर ऑफ आइसोलेशन एंड एंटीजेनिक सबटाइप H1 है, HA एंड न्यूरामिनिडेज, मतलब हीमोग्लूटिनिन एंड टाइप कौन से है. तो अब जैसे ये है डक से आइसोलेट हुआ है, दिस वाज द वायरस टाइप ए फॉर एग्जांपल. फर्स्ट वाला देखिए, ये दिस इज एनिमल ओरिजिन. तो टाइप ए डक, अल्ब्रेटा वाज द प्लेस ऑफ ओरिजिन, serial number was 35 and it was isolated in the year 1976. Type was H1N1 or the virus is from duck origin. अब ह्यूमन है तो ए, ह्यूमन में हमने नहीं डाला, एच नहीं डाला, यहाँ पर एच नहीं डाला, यहाँ पे डायरेक्टली हमने प्लेस ऑफ ओरिजिन डाल दिया. अगर ये कुछ नहीं है तो दैट मीन्स इट्स ह्यूमन. ए ऑब्लिक पर्थ, पर्थ से आइसोलेट हुआ है. सीरियल नंबर वाज 16 in 2019. Strain was H3N2 for virus from human origin. So in this way they are labeled. ये सब दूसरे examples हैं. So this is the actually epidemiology of timeline of influenza type A pandemics. अब इसको आप देखो कि ये स्टार्ट कहाँ से शुरू हुआ, मतलब कहाँ तक हम लोग इसको ट्रैक कर पाए हैं. ये तो रेट्रोस्पेक्टिवली ट्रैक किया होगा. 1890 में H2N8 का पैंडेमिक था. फिर पैंडेमिक देन 10 साल बाद 1900 में H3N8 वाज प्रेवलेंट स्ट्रेन. 1918 से 20 तक जो स्पेनिश फ्लू था दैट वाज टेरेबल. That was really, and इसमें कम से कम almost 50 million people were killed in this H1N1, right? फिर उसके बाद में 57 फिर चलता रहा ये, फिर उसके बाद में 1957 तक ये H2N2 एशियन फ्लू था। देन फिर 1968 में H3N2 हांगकांग फ्लू एंड 1977 में H1N1 रशियन फ्लू। So if you see the character of H antigen, तो from पहले H2 H3 हुआ, फिर H1 हुआ, फिर से H2 हुआ, फिर H3 हुआ, फिर H1 हुआ। तो next pandemic will be most probably because of H2. So in this way the prediction can be done कि ये H2 का, then we will correlate with the H2 pandemic कि उस समय कैसा था, क्या था उसका, उस समय कैसे कंट्रोल किया था। तो all control all the strategies to prevent this infection can be make out. So epidemiology is very interesting. I think you should go and read, and even the PSM people will also teach you about the. But it is also nicely given in the microbiology book how this influenza pandemic occurred in the last century, right? The Asian flu, Hong Kong flu, various names are given. Spanish flu. स्पेनिश फ्लू ने तो अमेरिका को जितवा दिया, ये शायद पढ़ा भी होगा आपने। स्पेनिश फ्लू जब हुआ तो स्पेनिश फ्लू होने के बाद में दे स्पेनिश पीपल दे डेवलप इम्यूनिटी अगेंस्ट, क्योंकि आपको कुछ समय होने के बाद में देन यू डेवलप इम्यूनिटी अगेंस्ट द इन्फेक्शन। नाउ अमेरिका वाज कॉन्कर्ड बिकॉज ऑफ दिस स्पेनिश फ्लू। विदाउट without firing a single bullet they won the America. इन लोगों ने वहां जाकर जितने वहां के नेटिव इंडियन जो कि नेटिव अमेरिकन थे, दे उन्हें कहा जाता था रेड इंडियंस, उन्हें जाकर ब्लैंकेट में ये इन्फेक्टेड ब्लैंकेट जाके उन्हें गिफ्ट कर दिए, और वो सब के बिकॉज दे वर नॉट इम्यून टू दैट डिजीज स्पेनिश फ्लू। तो धीरे धीरे करके वो सारे खत्म हो गए और इन्होंने अमेरिका जीत लिया। तो बायोलॉजिकल वॉरफेयर ये आज की बात नहीं है, ये पहले से भी ये लोग यूज करते आ रहे हैं। अब ये सब एपिडेमियोलॉजी है। द इन्फेक्टेड ह्यूमन आर द मेन रिजर्वायर ऑफ इन्फेक्शन, समझे? तो ये फॉर इन्फ्लुएंजा ए वाइल्ड एक्वेटिक बर्ड यूजुअली डक, दे आर वेरी इंपॉर्टेंट। डक से बर्ड्स में आता है एंड बर्ड से फिर स्वाइन में आता है, and in the bird they are in the intestinal epithelium, so they secrete the virus in the feces and contaminate water, ponds and lakes. अब इसको यहाँ से छोड़ देते हैं। These are the receptor जो मैंने आपको पहले बता रहा था कि sialic acid receptor, there is a two types of receptor. One is alpha 2,6 sialic acid receptor specific for the human influenza strain found on the upper respiratory tract epithelium. So these are the present. This is specific for the human influenza, right? So it will affect only the this 2,3 sialic acid. This is present only in the human. 2,3 sialic acid receptor specific for the avian flu strain found on the bird's intestinal epithelium and few on the human upper and lower respiratory tract infection. So that's why the avian flu occur in human, those who are in the close contact, right? But the human to human transmission does not has not been seen in the human because of few receptors of avian influenza are also present here. But both these receptors are present in the pig in abundance. So that pig will be infected by avian strain also and human strain also. And swine flu is also there. So the chances of genetic reassortment is much more higher in pig. So, so this is why the pig is the most common. Pig have both type of receptors. Swine flu contain a strain have a specificity for both receptor. Pig can be infected by human, swine, and avian simultaneously. Reassortment between segment can take place inside the some swine flu cells, swine cells. So these are the why the pig act as a reservoir, and they are the very important in the pandemic. This is epidemiology. इतना नहीं पढ़ाएंगे आप. Influenza is just like a minor respiratory disease, and इसमें क्या साइन एंड सिम्टम हो सकते हैं. साइन एंड सिम्टम्स इन्फ्लुएंजा शायद आपको सभी को हो चुका होगा. सीजनल फ्लू किसको नहीं हुआ है. रनिंग नोज, सोर थ्रोट, बॉडी एक, हेडेक, fever, tiredness, joint pain, sometimes gastritis, vomiting. These are the basically symptoms of mild flu. Everybody gets it and recover within a week. So only sometimes this infection can lead to the pneumonia, and this pneumonia is secondarily infected by bacteria, and then it is dangerous. So similar, just like corona, this infection start with this, but it does not affect the other thing like corona, right? So this is a very self-limiting thing, but if it occur, I mean, lecture. Complication usually rare in the primary influenza, but severe can occur. Secondary bacterial infection is one of the most important complication, which cannot be cured by uh, antibiotic because it causes the interstitial pneumonia. Staph, Haemophilus, influenza, pneumococci, these are the organisms. This is very dangerous in the elderly patient, and uh, elderly patient with debilitated or chronic lung disease, immunocompromised patient, and pregnant women. Right? CNS complication, Reyes syndrome, sometimes also seen. Now coming to the laboratory diagnosis. Now laboratory diagnosis जिस तरीके से हम कोरोना में लेते हैं, नजल स्वैब लेते हैं, नजल स्वैब से आप क्या देख सकते हो, एंटीजन देख सकते हो. तो एंड व्हाट इज द एंटीजन देखने की, व्हाट इज द मेथड्स, आइदर या तो इम्यूनोफ्लोरेसेंस और इलाइजा. दीज आर द टू थिंग. नासोफेरेंजियल, देन नासोफेरेंजियल स्वैब जो आप ले रहे हो उसको आप वीटीएम में जस्ट लाइक इन द कोरोना वी टेक द स्वैब इन द वीटीएम and we can culture it, right, in the monkey kidney or the baboon kidney culture. Third is we can see we can do the RT-PCR or the genomic uh, detection of influenza virus. So या तो हम direct specimen में हम antigen जैसे nasal swab first specimen, what specimen we will collect. तो nasal swab है, throat washing है, sputum है, throat gargle, viral isolation and serum for viral antibody. Right. So first is या आप लोगे क्या है में. So it's a virus. So viral transport medium is the suitable thing to be transport this. So just like coronavirus, you have to collect it and store it at 4 degrees centigrade up to 48 hours, and if not, then minus 70 degrees centigrade. If this is the VTM, this is the swab, nasopharyngeal swab, and this is the VTM. So just take the swab and put it in the VTM. This is the triple package. Triple package system should be there whenever the pandemic occur and organism or the virus is highly infectious. Then triple packaging system, it should be transported in the triple packaging system. देन लेबर डायग्नोसिस में हम फर्स्ट, मैंने आपको भी जस्ट बताया कि डायरेक्ट एंटीजन डिटेक्शन टेस्ट, आपने थ्रोट स्वैब लिया या नेजोफेरेंजियल स्वैब लिया, डायरेक्टली मेक द स्मीयर एंड you can directly detect the antigen by immunofluorescence, or you can direct ELISA method. You can, by using the monoclonal antibody, you can detect the antigen. So direct detection is either through immunofluorescence or by ELISA. Isolation of virus. तो आपने नेजोफेरेंजियल स्वैब लिया, VTM में ट्रांसपोर्ट कर दिया है, 4 डिग्री सेंटीग्रेड कोल्ड चेन मेंटेन की, उसके बाद में लैब में आपने भेज दिया। लैब में if the cell culture facility is available, then you can culture it on uh, cell culture. Previously, in the past, they used to culture it in the embryonated egg, that is amniotic cavity and allantoic cavity. But these are the blind procedure. Nobody does it. It is all outdated. And just for the theory purpose, you have to remember that the influenza virus is primarily isolated in the amniotic cavity and then in the allantoic cavity. But nowadays, only cell cultures are being used. All these are outdated. Amniotic cavity, I'm not going to teach all these things. Cell culture, monkey kidney and baboon kidney cell line, and growth इसमें कैसे detect करेंगे? Heme adsorption से या immunofluorescence direct. We will just add the RBC. If the antigen is there, so RBC get adsorbed onto the cell surface if the virus. So directly you can detect, or by immunofluorescence, right? Serology, fourfold rise in the IgG antibody will indicate that. ये भी कैसे detect करेंगे? All these complement fixation test and heme agglutination, radio immunodiffusion, they are all outdated procedure. Only ELISA and chemiluminescent assay, these are done. So just remember ELISA. Otherwise, all these are historical importance: complement fixation test, heme agglutination inhibition, and radial immunodiffusion. Molecular diagnosis is the recently the most important one, and we can detect this, and within 3 to 4 hours. For RT-PCR, just like in corona, we can detect the influenza virus RT-PCR, the nucleic acid in the nasopharyngeal. It can be a quantitative also. Treatment, usually we give amantadine. This was previously, but nowadays oseltamivir, zanamivir, or the oseltamivir, that is known as Tamiflu. These are the treatment available for the influenza. Vaccines are also available. There are inactivated influenza vaccine. It was grown in the cheek embryo and inactivated by a formalin. So this is not used nowadays. Cell culture vaccine is important. Made in Derby canine kidney, that is MDCK cell line. The vaccine prepared from the cell culture is used. Recombinant, this is most commonly used, that is recombinant influenza hemagglutinin antigen. Right? इसमें एक चीज और मैं बताना चाहता हूँ इसमें कि जो अगर कोई पेंडेमिक हो गया, अभी इन्फ्लुएंजा का पहले भी पेंडेमिक हुआ था, इन्फ्लुएंजा का अब नया पेंडेमिक हो गया, स्ट्रेन चेंज हो गया, सब कुछ हो गया, जो न्यू स्ट्रेन होता है पेंडेमिक का, इट इज वेरी डिफिकल्ट टू ग्रो और कल्चर दैट स्ट्रेन. इट टेक्स टाइम, राइट? So it is very difficult to prepare the vaccine against the new strain. तो इसका alternative क्या किया? Now we use the structure, the virus of the previous strain, just change the hemagglutinin and neuraminidase. So just hemagglutinin and neuraminidase of the new strain, and structure, the other things are of the previous strain. So it can be easily cultured. So this is important in the influenza. So nasal spray, one is the recombinant influenza, nasal spray available, nasal vaccine, live attenuated vaccine. So temperature sensitive mutant strain used. These aberrant strain able to grow at the 30 to 35 degree in the nasopharyngeal mucosa, but not at 37 degree in the lungs. So vaccines are available. It will give you protection for just a seasonal, that is 1 year, not more than that. Next year again you will have to. These are the current vaccines. So influenza H1N1, I told you about how this was uh, bird flu was. This H1N1 was the swine flu, and H5N1 was the bird flu, avian flu, that is H5N1. So avian flu virus multiply in the intestine of the bird, disease born. So basically, this avian influenza is restricted to the workers which are related to the poultry farms and other things. Most commonly is the type A H5N1 and H7N9. These are the two strain of avian influenza. So some of the incidences of outbreak of such are seen in Netherlands, Hong Kong, China, even in India in 2008, 9, and even in some pocket the incidences of bird flu is seen in India also. So avian influenza resistant to amantadine and ramantadine, but oseltamivir, that is Tamiflu, can reduce the duration. All human pandemic strain have originated by reassortment, that is genetic reassortment between avian and human strain, and pig has acted as a mixing vessel. So that's we have already discussed. Influenza surveillance के लिए the global influenza surveillance and response system is there. They monitor the evaluation of influenza viruses globally and serve as a global alert mechanism for emergence of. That's I told you that. In India also there is a surveillance system that if any outbreak occur, we have to report to the government. So there is a surveillance system and it is recorded in the system. Thank you very much. In this H1N1 now add Corona also. So we will see the more antigenic shift in the corona, and every 10 years we will see a picture like this. So thank you very much for your patience listening. Hope you were listening. Thank you.
Thank you, sir.
Any question? So if there is any question, then we will discuss it in the second lecture, that is on the paramyxovirus. Okay. So thank you very much once again.